This is video 1003, and this is on section 10.3, regulating the cell cycle. So here are our section objectives. Describe how the cell cycle is regulated. Explain how cancer cells are different from other cells. So how do cells know when to divide? Well, there are some controls for cell division. And these controls on cell division are called regulatory proteins. And they can control what happens both inside and outside the cell. Go back. So for example, when an injury such as a broken bone occurs, cells are stimulated to divide rapidly to start the healing process. The rate of cell division slows when the healing process nears completion. Because you know that your bones grow at different points in your life, and they enter what's called G0, and when they don't need to grow. And then if there's an injury, then there are proteins, regulatory proteins, that tell the cells to start dividing again. So these controls on cell growth and division can be turned on and off. So cyclins are a family of proteins that regulate the timing of the cell cycle in eukaryotic cells. So if you look at this, look at G1 phase. Remember, that's when the cell grows. So at that time, cyclin D is pretty high, and so is cyclin E. Then as it goes into S phase, cyclin E kind of goes down. Cyclin D is rising still. Cyclin A is starting to increase, and so is cyclin B. Now in G2 phase, cyclin A is pretty high, and then in mitosis, cyclin B is pretty high. So they all have different phases or different parts of the cell cycle where they are important. So this graph shows how cyclin levels can change throughout the cell cycle. So for instance here, mitosis, the cyclin concentration is high, and then it goes down in interphase, starts going up in mitosis, down again in interphase, and then up into mitosis. So one type of regulatory protein are called internal regulators, and they respond to events inside the cell. And these allow the cell to proceed only once certain processes have happened inside the cell. If you're just looking at this, if you do it on your notes have the link for the cell cycle animation. If we're in class, we will watch this, and I'll show you how the cyclins work. Now you have external regulators that are proteins that respond to events outside the cell. And they can direct cells to speed up or slow down the cell cycle. For instance, there are some proteins that are on the cell membrane, and once they touch another cell, it tells the cell to stop growing. And that's kind of how cells get to be all about the same size if you think of like plant cells. There are also regulatory proteins called growth factors. These are a type of external protein or external regulator, and they stimulate the growth and division of cells, especially if there is a wound, healing for a wound, or if it's an embryo that's developing. Apoptosis is a process of programmed cell death. So this happens when a cell is no longer needed. So it's lived its usefulness. And basically what happens is that the cell starts to shrink and it implodes on itself. You have what's called a nuclear collapse, so the nucleus collapses. And then you have lysis of the apoptotic bodies. Okay? So it would start here, and if you want, you can stop here and read what it says for each of the steps for apoptosis. You don't have to know the steps, just understand that there is a program, and some call it cell suicide. Here's an example of apoptosis or apoptosis at work. So obviously cells had to stop growing. Don't worry about the black and the pink. But on this mouse's foot, the cells have to stop growing, otherwise you would have one solid foot. You wouldn't have the individual toes. So how do cancer cells behave differently from healthy ones? If you click on this link, you will get to see a little bit of it. I'll show this video in class. Now, how do cancer cells differ from other cells? Look at the difference in shape here. Okay, and there's normal cell, and there's a irregularly shaped cancer cell. So cancer is a disorder in which the body cells lose the ability to control cell growth. Cancer cells divide uncontrollably to form a mass of cells called a tumor. So here's cancer in situ on the right and a benign tumor. Usually a benign tumor is enclosed in a membrane or a capsule, and then cancer in situ means cancer in tissue. And if you know anything about cancer, benign usually means that it's non-cancerous, and then malignant means that it is cancerous. So usually cancer in situ would be malignant. All right, here's a comparison of a normal cell undergoing what's called necrosis. Necrosis is when a cell is injured and it cannot recover from this injury. And what ends up happening is that it gets inflamed and the plasma membrane breaks down, and the nucleus breaks down, and then the contents of the cell leave. Whereas an apoptosis of a normal cell, that's with the programmed cell death, and basically it's phagocytosis of the cells, so the cell will eat another apoptic cell. Here's a picture of a benign tumor, and this would not spread to the surrounding tissue. A malignant tumor, as I said earlier, is cancerous, and it can spread. Another name for spread of cancer cells is metastasis. Now, this type of tumor can destroy surrounding health tissue. It can metastasize. In other words, it can go spread to somewhere else, and it can absorb nutrients. So it kind of has a blood supply in that it is taking nutrients up that other cells need. And it can also block nerve connections. And if it gets large enough, it can prevent other organs from functioning. So that's kind of what cancer does. So what can cause cancer? Well, usually it's caused by defects in genes that regulate cell growth and division. And I don't know if you know this, but do you remember what defects in genes are called? That's right, mutations. And mutations that lead to cancer are called carcinogens. Okay, so a mutagen would be something that causes mutations. Carcinogen is something that causes cancer. I bet you can probably name some carcinogens. Smoking tobacco, that's right, radiation exposure, defective genes, and a virus infection. Some of you may have had the HPV vaccine, and that is one that's called human papillomavirus. That's one that causes a type of cervical cancer. Defective genes, that could be caused from a number of things. Can be from the sun, can cause ultraviolet, the ultraviolet rays can cause mutations in your skin genes, or it can just be one of those things and it just comes up. Here are examples of skin cancer. If you look at the moles, they don't have clean borders, they're not even throughout. Okay. The main treatments for cancer are going to be surgery to remove the tumor, radiation to shrink the tumor. It's kind of like really, really strong x-rays they use to shrink the tumor. Chemotherapy, and that's when they use chemicals, and those are injected into the veins of the person, and that's kind of a type of treatment that goes throughout the whole body. So here's a little review about cancer cells. So exactly what is cancer? It's uncontrolled growth and division of cells. It's when the cell cycle fails. Cancer can crowd out normal cells. Cancer cells spend less time in interphase, so they divide pretty fast. If you remember from the POGIL we did last week, it mentioned that. It's caused by mutations in DNA, or it can be caused by environmental factors. Examples of carcinogens are asbestos, tobacco, secondhand smoke, UV radiation, X-rays. And sometimes there's families where the risk runs in families. It does increase with age. But it does require many changes in DNA. That's why it takes years and years and years usually for cancer to develop.